So we're glad to introduce our next speaker, Lorenzo Pasquini from University of California. He will speak about time-varying functional connectivity of the anterior insula in frontotemporal dementia. Thank you, Lorenzo.
Thanks for the kind introduction and for everybody being here. So in my research, I'm interested in a family of related neurodegenerative syndromes that fall under the umbrella term of frontotemporal dementia. And what the syndromes have in common is that, depending on where the atrophy is found, whether in the insula or in the temporal pole or in the inferior frontal gyrus, these atrophy patterns can somehow relate or can tell you which symptom the patient is going to manifest, whether it's a behavioral syndrome, a language syndrome, or a movement syndrome. And I study one particular syndrome, which is called behavioral variant frontotemporal dementia, in short BVFDD, which is a commonly found syndrome in early onset dementia. And the symptoms that these patients develop are quite dramatic behavioral deficits, where they lose, for example, they show loss of empathy, social disinhibition, apathy, compulsivity, and overeating. And what is almost very often found in these patients is atrophy of particularly the frontoinsular, also what I call here the anterior insula, and also of the anterior cingulate cortex. And as you can see here with disease severity, here with lower disease severity, these regions are already targeted by gray matter atrophy, and as the disease advances and the level of clinical severity advances, then we find even more widespread atrophy also in neighboring regions, and the atrophy is peaking within these two regions, the anterior cingulate and the anterior cingulate cortex. And these regions, and especially the atrophy regions that you find in BFDD, overlap very well with the silence network, a large-scale system that comprises exactly this region on the insula, the anterior cingulate, but also subcortical regions such as the amygdala and the periaqueductal gray, the ventral striatum. And this network has been involved several times with sustaining social-emotional functions, such as, for example, emotional empathy through its cortical, limbic, and subcortical structures. And while static connectivity measured through conventional connectivity approaches that use the whole time series in resting state fMRI have already shown that the silence network is characterized by reduced connectivity in patients with BVFDD. We now have more nuanced methods, as introduced by Vince, where instead of looking at the average static snapshot, we can see how connectivity within the system is changing at specific time windows. As you can see here, for example, where this connectivity here is maybe decreased at a specific period of the resting state acquisition, or maybe even inverted in connectivity signs, or where even nodes switch then to different modules through the whole acquisition of the data. And interestingly, previous groups have already attempted at mapping time-varying connectivity of regions of the silence network. Most notably, Naomi and colleagues have looked at time-varying connectivity of different insula subregions here, the dorsal, the middle, the ventral, the posterior insula, and we're able to find 5 states in healthy subjects and normal subjects where there were overlapping patterns of connectivity, but also where each state had a specific connectivity. We're showing specific connectivity to silence network regions, to the default mode network, but also to sensory motor areas. So also, we wanted to extend these findings by first looking how time-varying functional connectivity is behaving in regions that are specifically atrophizing BVFDD in healthy older adults, and then see whether time-varying connectivity in these regions are impaired in patients with BVFDD. And also, since there is a lot of uncertainty on what time-varying connectivity is reflecting, whether the time-varying connectivity changes are somehow related also to static connectivity changes found in patients. So we used 3 samples here for the study: a normative sample of a large sample of healthy controls, 121 subjects, a second sample of 23 healthy controls, older adults, and a third sample of patients with BFDD. All samples were comparable in terms of age. Actually, these 2 samples here were matched for age and gender and had also comparable measurements for head movement, which, as Vince also already introduced, is a very important confounding, not also for time varying, but static connectivity analysis. And so the first thing that we did was to investigate in our sample, where do we find atrophy? So can we just see what many people have found before, that if we compare patient gray matter between patients and controls, we would find reduced gray matter exactly in these regions that we expect it to be in anterior cingulate and in the frontal insula. But what this analysis also revealed is that the peak atrophy level was found in a cluster in the left ventral anterior insula, and this finding then also had an impact on all our following analysis, whereas we, for example, also looked at whether we could replicate previous findings of static connectivity decreases, showing that in BVFDD, the insula has lower connectivity to the anterior cingulate, this typical silence network reduced connectivity, which is found in patients with BVFDD when compared to age-matched controls. And we then proceed to the application of our time-varying connectivity pipeline, which was a variation of the sliding window approach, where we extracted a time series from the ventral insula, and then we built windows of 36 seconds length, and on a 2-second shift basis, we just slided these windows and built segments or periods of this time series of the insula. And then using a seed to whole brain approach, what we ended up was for each subject of the large sample, a set of 218 time-varying functional connectivity maps, where in red you can see positive correlation, in blue negative correlation, that indicate the level of functional connectivity of these regions just to our seed regions, to the left ventral anterior insula, to this kind of atrophied and heavily, this epicenter of the disease in our sample, kind of. So here you can appreciate a video for one subject of the time connectivity maps, where you can see that we have, for example, here on the lower left side, where we see patterns of, for example, positive connectivity spanning these typical regions of the salience network, but also negative connectivity in visual and somatosensory areas, then reverting to positive connectivity, and also like more frontal and orbitofrontal connectivity patterns. And so each of these maps was vectorized. For example, here this row is exactly this map here, and concatenated across the 218 subjects, and the K-means clustering with a solution of 4 was applied to derive 4 configurations or 4 spatial configurations that I will from now on call modes. And also the output from the K-means clustering was used, so how each of these windows was assigned to one of these 4 modes gave us a time series that we could use to tell how much time actually a participant spent, or the left ventral insula of a participant spent in mode 1, mode 2, mode 3, or mode 4. And here you can appreciate better the spatial maps derived from this approach, like the 4 modes, from the sample of 121 subjects, and you can see that there were overlapping connectivity patterns, especially within the insula, but also within the anterior and mid-cingulate, but they differed also in many aspects, like this mode 1 overlapped very much with a more kind of single-opercular task control network involving also dorsoparietal regions and also lateral prefrontal areas. And mode 2 was characterized by marked anti-correlations to primary sensory regions, in particular the visual cortex and the somatomotor cortices. Mode 3 had an inverse pattern of correlation with hypercorrelation to these primary sensory areas, and also anti-correlation to subcortical areas, which here were, as you can see, very, very, like the warm color indicate positive correlation. Then we had this mode 4 that was characterized by a more kind of ventral pattern of connectivity involving ventral salience network regions as the subgenual cingulate and also the orbitofrontal cortices. And we did several replication analyses with different samples of different ages, different K-means clustering solutions, looking also how the time spent in different modes is replicable. For example, in a 6-month span, what is the involvement of eyes closure in the data? What happens with global signal regression? If I could raise your interest, come and see me at my poster after the session. And anyway, so we proceeded then through the smaller sample of control and patients the time-varying connectivity maps that were generated for these samples were assigned to one of these four modes based on the spatial similarity, which here was assessed through Euclidean distance. And here you can see the templates derived from the 121 subjects, the healthy control, the smaller sample, and the VVFTD sample, and you can see that we were able to find very similar modes, and all three, and also the other two samples, also indicated by the high spatial similarity here measured through Pearson's correlation coefficients. So our next question was whether patients do and differ from controls in the time that they spend in each of these four modes. And while the time spent in this single opercular system and this anti-correlated primary sensory system did not differ between the two groups, we found that patients spend significantly less time in this hypercorrelated primary sensory mode and spend more time in this ventral salience network mode. And we then used a qualitative approach also to assess whether there were differences in connectivity within the modes. And here I'm just showing subtraction maps, and we didn't perform any statistics, also because one of the big problems that the field of time-varying connectivity is facing is which is the right statistical model to use when comparing different time-varying connectivity matrices or maps across groups. And I'm more than happy to talk with anybody who's interested in this subject later on. So, but what you can see is just through this qualitative assessment is that patients here, so red indicates higher connectivity in controls. So what you can see is that patients showed reduced levels of connectivity in this mid-cingulate, but also anterior cingulate regions of these modes. And we found this reduced connectivity here in mode 4 particularly interesting because if you remember the static connectivity changes that you can see here on the left, and here on the right you can see the overlap in violet. There is a convergence on the static connectivity changes, an anatomical convergence relating the static connectivity changes to these reductions in connectivity that we found in mode 4. So as a final piece of this analysis, what we seek to understand was the relationship between static connectivity and time-varying connectivity dysfunctions. And so we extracted average levels of connectivity between the insula to this cluster in the anterior cingulate just by taking the average beta values, which we plot here on the y-axis, and plotted this against the time that patients spent in this mode 4. And as you can see here, the negative correlation suggests that the more the patient spent in this mode 4, the lower actually the connectivity between the insula and the anterior cingulate cortex. So the more the left ventral insula spends connected to this mode, the more functional impairments you can find in the patients. So in conclusion, our findings suggest that the left ventral insula, anterior insula of patients spends more time connected to ventral portions of the salience network, but less time connected to primary sensory areas. And this is not trivial, since another alternative hypothesis would have been that since the insula and anterior cingulate are atrophied, that the insula gets input then from, for example, extra salience network regions, primary sensory regions, or even other cognitive networks, also DMN or attentional networks. What our findings also suggest is that while patients with BFDD spend more time in their kind of home-based network, in this ventral salience network, this time that they spend more in this network goes ahead with dysfunctional connectivity reductions within the salience network. So I think we need still to do a lot of work on this project. We haven't found a measure to assess whether patients with BFDD actually show a reduced range of mode occupancy, or if only it involves like these two modes that I have presented now, the hyper-correlated sensory mode and the ventral salience network mode 4. I'm very interested and eager to look how this time-varying connectivity deficits, the mode occupancy relates also to atrophy of the salience network, of the insula, of the anterior cingulate, in terms of both gray matter, but also DTI, structural disconnectivity, and also with behavioral symptoms like with clinical severity or with empathy deficits. And it would be also interesting to maybe scale up such an approach to study other disorders where you have insular dysfunction, where you have social-emotional dysfunctions like major depression or autism spectrum disorder. So I'd like to conclude here, and I acknowledge particularly my mentor, Bill Seeley, and my collaborators and colleagues at the Memory and Aging Center. A big thanks to the patients, to the family members and participants of the study, to our funding sources. And if you'd like to hear more about our work at the Seeley Lab, come to my poster later, or Jess is going to speak about some new approaches using convolutional neural networks to look at time-varying connectivity using the Human Brain Connectome dataset. And Salvo on Wednesday is presenting some interesting findings that combine neuroimaging in patients with TFTD with post-mortem neuropathological data. Thank you very much.